Uh, Good evening, all. First, I would like to thank the organizers for giving me this opportunity. And I'm Rajeswari Appadurai from a postdoctoral fellow working with Dr. Anand Srivastava's group in Molecular Biophysics Unit of IISc. And I'll be talking about my latest work on intrinsically disordered, latest work on dimensionality reduction methods applied to protein ensemble data. So first of all, to introduce to what are protein structural ensembles and why are they complex to study. So proteins are long polypeptide chains composed of several thousands of atoms, and for describing one particular structure of a protein, you need 3n coordinates. And these protein structures are not static in the cell, and they adopt a range of conformations in the solution, and thereby they can take multiple structures. So to describe all these ensembles, you will need a better collective variable upon which you will plot their free energy landscape. So the free energy landscape also can be very diverse based on the nature of the protein. Certain proteins like folded protein certain proteins like folded proteins are existing in the funnel-like landscape with favoring only the native folded structure, whereas certain other proteins can transform between two or multiple conformations, which are rapidly converting in an equilibrium setup. So from the process of sampling to the visualization of this landscape, it all depends mainly on the order parameter that you are choosing for representing these landscapes. So if you think about the classical collective variables or the order parameters like RG or the RMSD, you can get completely different structures for the same RG value or the same order parameter. So using these order parameters can be a poor choice for representing your free energy landscape. So people are now mostly using the dimensionality reduction methods for identifying the latent features of the protein structural ensembles. So there are many dimensionality reduction methods are applying. Most of them are linear and non-linear based methods. So principal component analysis is a famous example of linear method, and for non-linear methods you have isomap, MDS, and TSNE methods. And in the interest of time, I'm not going in detail about the working principle of all these different methods, but what I wanted to study here is among these many different methods, when your high dimensional space to the low dimensional transformation is merely a black box, how do you identify which method that best suits your suits for representing your underlying data? So how do you choose that? So you need an automated framework for that qualifies the method for representing your data. So we are aiming to provide that, and we initially tries to take the framework that are already available in the neurocomputing field, and this method of quality assessment is based on distance ranking. So consider a two-dimensional original space, which I'm reducing it to a single-dimensional space here. So this method of quality assessment is based on calculating the distance ranking of each central points in the original space, with of the distance ranking of all other points with respect to each point in the original space as well as in the projected space. So the mismatch of these ranking errors are then counted and represented in the Coranking matrix. So in the Coranking matrix, what you are plotting here is what are the number of mismatches for a specific pair of mismatch in the low dimension as well as in the high dimensional space. So this Coranking matrix essentially tells you, given a cutoff region, what are the allowed mismatches, the soft mismatches, or what are the extreme mismatches in your projection with respect to the original space. The lower quadrant here will tells you about the highly squeezed points in your embedded space with respect to the original space, otherwise which are called as intrusions. Whereas the right upper portion tells about the extruded points or the stretched out points in the embedded space than in the original space. So with these Coranking metrics, these people, Lee et al., they have come up with different matrices called trustworthiness, continuity, and LCMC. for analyzing the effectiveness of your projection space. When we are applying this protocol on our protein ensemble data sets, we still see that the free energy map obtained from the dimensionality reduction method is not able to capture correct statistics. So then we have introduced two other metrics that are based on distance ratios. So essentially these metrics are counting the number of mismatches in the distance ratios from the low dimensional with respect to the high dimensional data set given a tolerance cutoff, and the other method is to evaluate the neighborhood preservation. So when you are projecting the low dimension and the high dimension, you are just counting what are the common neighbors in the low dimensional space over all the neighbors in the low dimension as well as high dimensional space that we are measuring here. So the overall overview of our workflow is following. You have the input data of atomic coordinates for all your snapshots, which we are converting to the distance matrix data using RMSD or Euclidean distance or cosine distance. So this distance matrix are feed into the dimensionality reduction algorithms, different dimensionality reduction algorithms we are applying. And then we are quality assessing it using the methods that I have just spoken about. And then based on the quality, we are using that projection techniques for visualizing and interpreting. We are also using this data for conformational clustering. So the first application of these framework is on the beta hairpin flow folded trajectory, where we have started from the completely unfolded state to the folded state. The sampling is guaranteed here. So with this trajectory, when we are applying different dimensionality reduction methods, we are considering only 5 methods here, but we are planning to apply other methods also. So for TSNE, there is a hyperparameter that you need to optimize. That also we are optimizing using the quality assessment framework. Just a minute. So when we are applying different methods for this data set, and we have checked their quality using the methods that I have described above. So the first column tells about the ranking based metrics, where you can find that the TSNE with perplexity of 5000 can capture the high dimensional space in a better way, whereas the distance based ratio is telling that the isomap is the best method out of all the different methods. So when we are plotting these two methods, the isomap and TSNE, when we are plotting the free energy landscape across these coordinates obtained from the isomap and TSNE, we find that isomap better represents the physical nature of the protein. The beta hairpin is a folded protein where you can see the folded funnel-like structure here, whereas the unfolded state is very broad and highly diverse. Whereas the TSNE, which come first in the ranking-based matrix, you can still capture the folded space with respect to the unfolded space, but the nature of free energy difference between these states are not captured correctly. So then we have applied this on the drip cage folding similarly, and then we find out that again isomap comes as the top in the distance-based ranking. But one thing we have noted in these data sets that when you are having the low neighborhood size, meaning the local space similarity measurements, you can always see that the TSNE is coming first. So we thought that okay, TSNE could be so good for preserving the local space than the global representation. So it seems like currently that the isomap better represents the folded protein, but we thought that this TSNE, which preserves the piecewise or local informations, can be used for some heterogeneous data. So heterogeneous proteins like IDPs, maybe in this set of proteins, we can use these. As I already mentioned, the intrinsically disordered proteins are very heterogeneous set of data, heterogeneous ensemble, and they are readily transform across multiple structures, and thereby they are allowed to perform multiple different functions. So you can think of IDPs as a Swiss knife, given each substrate of the structure with different functions. So in this set of proteins, you may not wish to sample. You may not wish to sample the global space because it is just a flat space. Rather, what you will like to get from this space, from these proteins are to get the different substructures of these complete ensemble heterogeneous structures. So that you can match their distinct functions of each of the substrate, or you can design for therapeutic purpose or anything. So one of the protein, IDP protein that we are illustrating here is the amyloid beta peptide, which is physiologically very important and therapeutically very important. And there are studies on this A beta. Thank you. So there are studies on this A beta peptide in the apostate as well as when it is binding to the inhibitor, which are recently published in this paper. So we have obtained these trajectories from this paper by Heller et al. In the apostate as well as in the ligand bound state. So in this paper, they have applied classical clustering algorithm like hierarchical clustering. But what happened is you can see the dominant cluster in both the apo as well as in the ligand bounded state. The dominant cluster corresponds again to the very heterogeneous mixture of data. The dominant cluster is not separated into unique states again. So when we are applying this when we are applying TSNE on this data sets, so we can see that all the conformational ensemble of the A beta in apostate as well as in the ligand bound state can be categorized into distinct homogeneous states. So in each of the state, you can see the secondary structure is intact, unique for each of the substate. Not only the protein part, when we are doing this analysis with respect to the protein-based similarity matrix as the input, we can also see that the ligand is captured as in a more specific bind condition. So with this information in hand, we can construct the thermodynamic estimates more faithfully, or you can study the kinetics of these transitions from one state to another using Markov state modeling, or you can use these substructures for therapeutic design strategies as that of the conventionally like that of the folded protein, where you will need to find out the specific binding pocket or the specific residues to target for. So these kind of informations now will be clearly can come up with this kind of faithful classifications. So with this, I would like to summarize and conclude my talk that we have presented a unified framework for comparing the different dimensionality reduction strategies, and we have found that the TSNE can be utilized for faithfully representing the IDP heterogeneous ensemble data. So I would like to thank my collaborator Upal Bhattacharjee and my group in Anand Srivastava's lab, also my funding agency and computational facility in IISc and Compute Canada. Thank you.
So we have time for 2 questions. Is there a question? I wanted to ask you a question about the Markov state model classification. So typically what we do is you run a bunch of simulations, and then from the free energy landscape you try to sort of categorize them into different Markov states, right?
Yes.
So how exactly are you using the categorization here in this particular case? What are the cost functions, or what is associated here?
So essentially, whatever the formula that you are using for a typical molecular simulation trajectory, only the classification scheme you will use with this parameter. So anyway, when you are, so what I mean is when you are, the trajectory is like the normal way. Trajectory construction is by the normal way. After that, to find the unique state, you are doing it with the dimensionality reduction method. In that sense, you will capture the unique states in a better way. That's what. Yeah.
Thank you. Is there any other question? If not, please thank Dr. Rajeswari Appadurai.